Professor Jairo?
Hi.
Hi, Professor Jairo. Am I audible?
Very nice to meet you.
Yeah. A very early good morning to you from your Colombia and a late good morning from my side from India. We are very delighted to have you, and I'm very thankful to you for uh, your presentation, which you would be further presenting in front of all the 450 participants with our international conference being held for 2 days. And now I would just um, pass on the system to Nikita, final semester biotechnology student, for a brief introduction of our eminent speaker from Colombia, Medellin. Yes, Nikita.
Good morning, everyone. It is my immense honor to introduce Dr. Quijano. Dr. Jairo C. Quijano is working as a professor in Colombian Polytechnic JIC, Medellin, Colombia. He completed his engineering and MSc in physics from National University of Colombia in 2004 and 2007, respectively. He completed his PhD from Swiss Federal Institute of Technology in Lausanne, Switzerland in the year 2013. He has published over 20 research articles in various peer-reviewed journals. His area of expertise includes cell and cancer biology, biophysics, and plant protection.
With this brief introduction, Professor, I would like to invite you for this talk, and you can share your slides and please present your presentation.
Okay. It's a pleasure for me to be here. I'm gonna to present my PowerPoint right now. Please confirm me if you can see it.
Yes, Professor.
Okay.
Yes, good to go. We can see the presentation.
Okay. So here we go. So first of all, um, I'm gonna thank the International Conference on Nurturing Innovative Technological Trends in Engineering for the invitation, and a big thanks to Dr. Ujwal for facilitating um, this like a virtual exchange of research interests. So I think I'm gonna turn off the camera just to
Yes, Professor. Yes,
yeah,
yes, sure.
okay, to focus on the presentation. So yeah, so okay, so my presentation is entitled UVC radiation for controlling gray mold disease in cut roses. Uh, the first thing to say is that this project was carried out in the company with the Tropical Plant Breeding Research Group here in Medellin, Colombia, as well. And I am the director of the Applied Physics group. So this work has been done with these two groups in collaboration. Um, Here we will have the opportunity to see mainly how different sciences are combined with engineering to bring like alternative solutions to some agriculture issues. In this case, for controlling fungi that is basically known as causing the gray mold disease. So I'm gonna okay, I'm gonna go for the first slide. So so I'm gonna have an introduction. an introduction, a brief introduction into the subjects that we touch here in this presentation. And the first thing is just to realize that the fungi are the greatest threat for food to food production in the world. All right, here we have images of tomatoes in a greenhouse, totally like yeah, messed up with some fungi. And traditionally, this kind of issues in the agricultural aspect has have been treated with synthetic chemical fungicides. Normally, that is the tradition worldwide. So we all know, or we can guess, that these chemical approaches are really invasive, not only with the plant itself, but also with the environment. And if you are not really careful, finally um, the people that operates these crops are affected too. So this is one main concern. The other concern is that this like
Professor, I'm sorry to disturb you, Professor, but
yeah.
we are not able to see the presentation if you have already started it. We are not
Oh,
able to see the slides, please.
okay. Mm, right now, right now you can see it, or
Yes, Professor, yes.
yes.
Perfect,
Okay.
perfect.
Okay. So again, you can see this image of a tomato greenhouse plantation. Um, okay. So I was saying we know that fungi is the greatest threat to food production. All right. This is treated normally with synthetic chemical fungicides that affects the environment, the human health, also the soil, and finally, we know that these fungi have the ability to develop a resistance to these chemicals. So this implies that if you're using chemicals, afterwards you need to like increase this the concentration of these chemicals, and this unfortunately is um, is like a cause for a public health issue and high risk for the environment. Um, this has forced the industry to search and develop alternative methods for disease management. This is mainly the cause for searching alternative methods. So one of these alternative methods is like a physical approaches. So there have been also other approaches like biological methods, and here we're gonna focus just in one physical method, which is the use of light. As simple as that. It's just using light. In our case, is using UVC light. So since we're gonna talk about a little bit of light, I can just explain some basic things about light. So light is an electromagnetic wave, and electromagnetic wave has an energy which is given by this formula, which is h, the Planck's constant, and nu, which is the frequency, or in other version, frequency and wavelength are inverse. So basically, uh, light is energy, all right? Is a wave that carry out energy, and is quantified by the wavelength. And Here we're gonna have just light interacting with matter. In this case, biological, meaning the fungi or the plant. So there is a propagation of photons that are the carriers of the light. And okay, think more about that. The important thing is to understand that the wide spectrum of electromagnetic wave, which is called light, which is not called light, but um, overall, an electromagnetic spectrum. Here we have it as a figure that can help us understand somehow the relationship between the wavelength, the frequency, and some images that we can be familiar to. So here we're gonna be in this region, the region of the wavelength of of more or less the size of molecules, and the frequency implies that the energy is really high, not as high as the X rays or gamma rays, but it's pretty high. And um, we can relate this to also compare it with another electromagnetic waves that are used in medicine, for example, like X rays or gamma rays, or even infrared are used too. So here we're going to focus on this, just a small part of the electromagnetic spectrum that is called ultraviolet. And even in the ultraviolet spectrum, we're going to focus just in the C part. So before that, we need to understand that a light or any electromagnetic wave can interact with matter in different manners. So basically two. There is light that can cause ionization of molecules and light that do not cause this. So here the ultraviolet, the UV light is just in the limit in between the energies that can cause photo ionization like X-rays and gamma rays, and the ones that do not do that, like the visible light or infrared or microwave, that is a common electromagnetic wave used in the for heating our food. Okay, so this means that this kind of wave, ultraviolet violet UV light, is not able, is in the limit of being able and not being able of cause some ionization. So this is a very very important aspect of this light. Okay, now focusing the UV, we can split this UV in three parts: UVC, which is coming from 100 to 280 nanometers; UVB, which is 200 between 200 and 315 nanometers; and UVA, which is the UV light, which is closer to what we call visible light, which is where are able to see as or we consider visible light. Okay, so here this picture shows that the UV light is mainly, the source of the UV light is mainly the sun. So this is an important thing to have in mind, because the ozone layer can let pass just a few UVA and UVB, but UVC, the ozone can absorb it. So mainly here on Earth, we cannot have UVC light coming from the sun. This is also important to notice the the high concerning about the ozone layer, right? Because the ozone layer is making this work of not letting this highly energetic light to come to us. So, all right. So that means that if we want to have an UVC light source, we need to produce it. So, how to produce UV? How does it work mainly? So we have sources of UVC light, like artificial ones, coming from just lamps, two different kind of lamps. Maybe the one on the center is produced by LEDs, and the one on the left is produced by a gas, so a lamp gas, or what is called a germicidal lamp. Okay, how does it work? So basically, it's kind of like the wavelength that characterize, sorry, this kind of light is really close to the wavelength that is affecting the DNA. So for any structure, DNA structure, this UV light can penetrate till the DNA, and it causes these ruptures in this kind of bonds. So this makes the DNA to form what is called dimers, and therefore the whole process of DNA polymerase and this enzyme cannot work properly. That is the direct effect of the UV, is directing, pointing the DNA structure of any organism. So this is how basically and briefly how the UV works. In particular, the UVC, which is the most important uh, light in order to produce this kind of effects in the DNA. Okay, now since we're talking and we're gonna talk about the roses, uh, we need to understand why roses, why are they important, at least in Colombia. So the importance of floriculture. First of all, here we have a chart in which exposes the world's largest cut flower export countries. So Colombia here in green is one of the main exporters of flowers in general, right? So this means that this is a really important income to our country. And in terms of roses, Colombia is really the first exporter worldwide. So it's important that Colombia exports a lot of other flowers, but the roses is basically one of the main entrance of income to our country, and in particular in the region that I work, with Medellin and the surroundings, are well known or worldwide known for this kind of flowers. So yeah, we were focusing on a big entrance to our region and some problems that these kind of crops have. So what are the problems really? Well, first of all, we need engineering. We need engineering applied to agro industry. Here in Colombia, uh, unfortunately, the agro industry of this kind of flowers, roses, is really like let's say old. The engineering applied to this kind of crops is really not much, and need to have a little implementation. So we have little implementation so far of emerging technologies. Here we have in the on the right typical UV lamp, which can be used and is already used in some developing countries. So to treat plagues. basically. So here we have the main goal is to evaluate the effect of this kind of radiation on the development of fungus. All right, that is the main concern and problem in a highly important crops in crop in this country, which is the roses. So here we have a basic method which consists only in a commercial lamp, UVC lamp, a camera to acquire just the images, uh, a segmentation program that we develop to measure the infection area, a program made in MATLAB, or it's a basic segmentation program that, that we developed, and we need a microscope with a common one, a wide field microscope, to just to see some like the germination of the fungus, and the okay, the fluorescence microscopy is just to check if this kind of light can produce internally or molecular, or I mean cellularly, some kind of if it can produce something. It's just to check that. So this is here I'm going to present a really easy methodology to follow this high and uh, really concerning problem in this industry. Um, I'm, I need to say that this kind of problems is are really important to this industry, because countries like United States, which is say the main importer of this kind of flowers, can put fines, economical fines, if the shipment of these roses uh, show this kind of fungus, right? So it's really important that to control this fungus, and how we control it, because lately uh, the importer countries that are checking also the use of chemicals. So it's really important to check if we can produce, or we can have a different approach to the chemicals. So this is why this kind of approaches, these physical approaches, takes importance. So basically, this is the segmentation program that we developed in the MATLAB platform. So here the goal was to just measure. Here we have an image of a rose with the fungus. So this region that is affected, we need to measure in order to have some quantitative results. So we just developed this program, as I said, in MATLAB, and in order to just measure this area, this lesion area, and in order to compare it with the total area of the petals, for example. So here first of all, we developed this software, and then we just apply these doses of UVC light, and we just try different incubation times. Incubation times meaning the incubation times of the fungus on the rose, because we can control the fungus. So we put the fungus on the petals, on the roses, and we just check. We give it, we have a control, which just means that no UVC light was applied, and we have a dose here in 1080 joules per square meter, and we just check how does it work. Okay, so we check just different incubation time. This was for controlling and taking care of the time that the fungus needs to to fully, let's say, contaminate the, the flower. All right, so which we here we can see that 24 hours of incubation time is is an incubation time in which the dose applied is really taking control of this fungus. But if we check 27 hours, it's maybe too late. It's maybe too late because the fungus is already here in the day 6. You can see it a little bit, but you can see it. Uh, for flowers, it's really important that a flower uh, is healthy. If a flower can have a little stain, it can be rejected. So it's important in this kind of product like roses, the appearance is really important. In fruits, maybe not, but in flowers, since we have to have beautiful flowers, these stains, little stains are really problematic. So, okay, here we have a chart which is resuming the images, the data coming from the images measured with the segmentation program. So basically, the conclusion here is that 24 hours of incubation time is a time in which we can see that the UVC light works better. In 27 hours is maybe too much time for the fungus. The fungus had fully uh, penetrate the roses, and therefore is more difficult. It's more difficult to control it. But even so, there is a significant reduction of the area of the lesion area. All right. Therefore, we choose these two times: 24 hours of incubation of the fungus and 27, and we apply just different doses, UVC doses. So here we have that for 24 hours, the 3 doses that we use are really controlling the fungus, and for 27 hours, really the 3 doses that we try was all right. They control a little bit the fungus, but not totally. So here we have at the bottom a chart that's resuming this, and we also try not only the fungus on the petals, but just the fungus alone. So, on petri dishes, we just grow this fungus and just irradiate with UVC directly, just to check that the effect is on the fungus and not on the petals. So here we can see with these yellow arrows the germinated tubes of the fungus. This means that this kind of fungus they are alive, they can germinate. And here in the B chart, we have the fungus with not able to generate these germination tubes, meaning that this fungus had been really controlled. So with this chart, we just check the direct effect of the UVC is just on the fungus, which is really a step that to be care, because first of all we have to just be sure that this UV do not affect the roses itself, just the fungus. So here we have this kind of experiment that really clarifies that. And this is the part of the fluorescence microscopy. This part, we have heard that maybe not only that this UV can control the fungus by meaning of affecting the DNA, but also that we heard that this UV in some previous works that the UV light can induce the plant to produce some kind of molecules that can protect the plant for a further invasion. And one of these molecules is called chios. So this chios deposition is a molecule that acts like a shield in the membrane, the cellular membrane of the cells, of the plant cells. And these formations, these chios depositions, as I said, apparently are induced by this UVC light. Here we have some experiments that show the brighted spots are chios. So after a UVC treatment, we can see some formation of this. So not only this light just have a direct impact on the pathogen, in this case a fungi, but also it can produce this kind of molecules that are like protective for the flower, in this case for an upcoming treat like a fungi. So this is like a second, like a secondary but a really important aspect of the treatment with this light, as a preventive, not only taking care of the fungus but also preventing for a following invasion of or any microorganism. And okay, finally we have like a prototype. We construct a prototype for using this. So we have like a UVC radiation prototype, which is a cage which we just put it in some sensors, UV sensors, movement sensors. And here we have a picture in which we have Rose just getting into the prototype. Here we have a view from the inside, and we create this kind of prototype because here we not only have like a summary of different sciences applied in here, but we also have like a formation of students. And here we had the opportunity to work with agricultural students, agriculture and engineering students, and some instrumentation engineering students too. So this was really nice because we have an interaction of multidisciplinary aspect of this project, and we can, with the instrumentation engineering students, we create this prototype. And basically we treat we prove this prototype and to be really useful and to be used afterwards in a large scale in the industry maybe. So as a conclusion for this kind of project, we have the effective control of this disease with a UVC, which is, again, is not invasive. It's just light, right? You need to take care a little bit of light, just wearing glasses and this, but it's by far, it's not dangerous at all compared to the chemical approaches that are traditionally used. And take advantage of the development we did and the combination of lights and images analysis for substitutes. Maybe we can use it as a substitute for traditional producers. Laboratory scale prototype was designed very low cost, just a couple of really cheap sensors, UVC sensor, and maybe movement sensor. That's it, no more than that. And this methodology shows that physical methods can be applied to agriculture and can be really reliable, environmentally friendly, and safe. And this is basically what we have with this roses project. But as I was saying before, not only the scientific contribution has made, just to taking care that take into account that interdisciplinary has been made, and we produce some papers, which is okay, is scientific results, but also the formation, as I was saying, thesis, undergraduate thesis in instrumentation and control engineering, undergraduate thesis in agricultural engineering. This is a great example of how uh, this kind of science can be applied in a such a problem which is roses, and we have different approaches, different views of different engineers to just combine those and have finally some um, great results. So we have some ongoing projects in the same thing. So now we have just like in a scallion on a scallion onion crops. We just we want to create, and we are trying this kind of prototype, but this prototype is on field. This is on field. This is not. This is a pre-harvest, so we can use this kind of like pre-harvest, post-harvest. Here we have direction crops, and we have also on bananas, just to control a hugely scary little fungus called that caused the crown rot disease in bananas worldwide. So we have here just another prototype, another project ongoing. Uh, with just, for example, here we have just the fruit. With the scallion onion, we're trying on crops. So we're trying to put this kind of approach in the middle of the process for the bananas, and we're just going with this since it's uh, has very like good results, and maybe can be applied to other kind of fungus and other microorganisms too. So that's all. I think I cover up all the mainly aspects of this project and the upcoming projects too. So I don't know if have some questions about it. And again, thanks for having me. Um, I'm ready for any discussion or question that you may have.
Yes, hello. Am I clear now?
Uh, Thank you, Dr. Quijano, for that wonderful and well elaborated talk. Can you hear me?
One.
Yes.
Okay.
Yeah. Yeah.
We have a couple of questions. One is from our faculty, Ms. Harshita, that does UVC affect the shelf life, color, and sensitivity of the flower?
Yeah. I didn't I didn't show that because it's a lot of slides. But basically, yeah, we did. We check this UVC light before, so just on roses, on healthy roses, just to see if the roses were uh, at the end. I don't know, maybe cooked by this kind of light. But we make sure that we have the doses that we cannot see any of, let's say, visual effects on the color, the shell life. We try to, and we have in our paper, uh, we expose this, and we say that we try the shelf life, and is some in some cases even prolong the shelf life. So this UV is really interesting. It's not only maintaining. I mean, if you need to try before what kind of doses are not burning this these roses before checking with the fungus, but even the shelf life has to prolong it a little bit. So yeah.
Yeah, thank you, Professor. I have a couple of questions. One is
Yeah.
Somewhere I remember reading that UV radiation at night is said to be more effective. What could be the logic behind it?
Sorry, I didn't hear it quite correctly before. There was
The
an
radiation,
interference.
UVC radiation, if it is
Yeah.
given at night time, is said to be more effective. What could be the
Okay.
logic?
Yeah, we here in this in this project we tried that too. But we try and we call it a dark, like a dark period of
Okay.
time. In our case, because we have heard that we read that on some previous works, there are differences, and in some works that this dark period after the UVC application and can like in some cases can help this the plant, in this case roses, but we haven't heard on roses. It's just in other plants, on other fruits, that can help this UVC approach, and some others didn't show that. In our case, we tried this dark period of time, like we tried 4 hours after, and basically what we found is that in our case it did not improve. It did not improve, but it improves, it's mainly they say that some researchers say that the plant has the time to recover this stress, because UV light is like somehow and stress for the plant. So the plant can recover for this 4 hour for this application of a dose, can recover the plant, and the microorganism may recover too, and this can add or not to the effect of the UV. In some plants, as I say, it works. Some doesn't. In our case, it did not work. I didn't show that because a lot of slides, but in our case, it did not. It did not work. It was like the same results. So maybe we need to use more like a dark period of time. I don't know, but yeah, this is what we found.
Okay. One more question from Dr. Smriti Prabhu, that
Yeah,
as UVC is not required for the plant growth,
yeah,
can quickly affect the plant's DNA or damage the DNA. Hence, how can its application be justified on field?
yeah. Basically, this kind of light, UV light, can go deep, right? Can go deep and affect the DNA chain, as I showed. The thing here is that there are different microorganisms. So this microorganism has different cellular membranes, right? So in our case, for example, we make sure that the rows were not affected. In our case, is let's say morphologically, like we cannot see stains on the plant, right? So the effect that is the DNA is being affected, and therefore the cell die, and this can be seen as a burnt let's say plant, in this case roses. In our case we didn't have that, so but we have that on the fungus. This is why we can use this, because this is affecting the fungus in a dose that only affects the fungus. It will affect also the plant, but with a higher doses that we checked before. We checked the doses that burn the the roses, but from that results we just reduce this kind of these doses, and we check if those doses can affect really the fungus, and this is what we have. So of course we first need to check on plants, on crops, first the VUC that cannot but did not induce this kind of effects of burning. And then we are ready to just show on this microorganism. But the microorganism takes this light differently because they can produce this kind of chills and that reacts differently. In our case, the petal, the roses, let's say cells, produce this kind of chill, which is called chaos, and protects them for this kind of radiation. Meanwhile, the fungus did not. So,
Yeah, thank you so much, Dr. Quijano.
yeah, problem.
I'll hand over the mic to our student representative.
Okay.
As we come to the end of this session, it is my privilege to extend gratitude towards Dr. Jairo C. Quijano for taking time out of his busy schedule and bearing with the time difference to deliver such an enlightening lecture on the application of UVC radiation in controlling the growth of pathogens. Thank you, sir.
No problem. It's been my pleasure. Thanks for the invitation. That's it. Thank you very much.
Thank you. Thank you once again, Professor.
No problem. Anytime.
Yeah, I think ಅವ್ರದ್ದಾಯ್ತು ಸಂದೇಶ್ ಅವರು ಎಂಡ್ ಮಾಡಿದ್ರು.